start here. Okay, so last time we were talking about inducible operons. So those would be operons that are off, but you turn on based on the need of metabolizing nutrients, like in the case of lactose. E. coli has the lac operon that once it has lactose available, it will start producing these proteins, these enzymes that help them to assimilate this lactose. Now, the next type of operon that we're going to talk about is the actually repressible operon. So this operon is always on, and you need to turn it off. Now, why will this operon will be on all the times? Well, it's because one of these essential amino acids, like tryptophan, is needed all the time. And then, no matter that you have this need of having on, sometimes you can turn it off. So, trip operon or TRP operon, it will be repressed when you have excess of tryptophan. That will be a co-repressor. So this co-repressor will bind to the repressor itself, and then when this co-repressor binds to this repressor, the RNA polymerase won't be able to bind into the promoter region so that these structural genes won't be expressed. So you stop the synthesis of tryptophan. So here we have an image of what will this operon will have, the tryptophan operon. So you have a regulatory gene that it is known as I, and then you have a promoter, an operator, which is a control region, and then you have A through E structural genes. And then when you have this operon working, you are always having the I gene on, so that means that you will have this transcription going on. And then you will have a translation for having an inactive repressor protein. This inactive repressor protein will bind into this operator, preventing the transcription of these genes when you have tryptophan. And then within this inactive repressor, you have a site where you bind the co-repressor. In this case, will be tryptophan. So then, since this inactive repressor protein is not binding to this operator, then the DNA polymerase can transcribe all of these structural genes, and then you will have polypeptides comprising of enzymes that will make tryptophan synthesis. So then, how do you turn it off? Well, if you put your bacteria in a media that is rich in tryptophan, or if this bacteria finds an environment that is rich in tryptophan, there is no more need to transcribe these structural genes because tryptophan is already available. So in this case, yes, you will still activate the translation of this I or repressor protein, but this will be active once you bind the co-repressor, which is tryptophan. So that's why you have this little site, and then this will change this shape of this repressor protein that will make this repressor protein now be able to bind into this operator, and then the the RNA polymerase, when it tries to bind to this promoter, it won't be able to bind to, because there is a physical distortion in here, or physical interference that will prevent these other genes to being activated. Okay, so here we have a question. So use the following metabolic pathway to answer the questions that follow it. So in this case, this pathway, you have a substrate A, that will be converted into intermediate B by this enzyme A, and this intermediate B will be converted into the end product, which is C, by the enzyme B. So if enzyme A, this one, is inducible and is not being synthesized at present, a blank protein must be bound tightly to the 2 blank. When inducer is present, it will bind to blank 3 so that blank 4 can occur. So let's fill in the blanks. So we are talking about an inducible operon on this question, and then the next one is going to be a repressible operon. So if you have an inducible operon, what would be the first blank that would fill in there?
Inducer protein.
Okay.
And then to the operator site.
And what would be 3? Present. 3 is present? No. Okay. When the inducer is present, it will bind to the blank.
They're repressive. Oh, yeah. Okay.
So that what can occur?
Transcription.
Transcription. Yeah. So everything is related to transcription, of course, right? So this is A. Then what about B? Let me put it this way. If an enzyme A is repressible, end product C called a blank causes the 2 blank to bind to the 3 blank.
One would be co-repressor, right? It binds because
So will be 2 what? Causes the inducible what?
inducible.
You can go back. Or inactive. It's inducible or inactive, right?
Mm-hmm.
Let's put it more accurate. Inactive. And then to bind to the operator. So what will cause the repression to end? What will happen with this? So what will make this repression to end?
The tryptophan would be released from the active repressor protein.
But yes, right? Tryptophan presence or it's called a tryptophan. I'm thinking about something else rather than only tryptophan. I mean,
Excess buildup of end product.
yeah. So it's just when you have the end product, it's like a feedback mechanism when you stop the repression. Okay, now let's talk about positive regulation. So in this case, we need to know about catabolite repression. So we have some molecules that happens after you metabolize an initial product, and then you end up with a waste or an intermediate. In this case, will be a catabolite, and that will inhibit the cells to using other source of energy. For instance, we have cyclic AMP that will build up in our cells, or well, in cells in general, when glucose is not available. So this is what sometimes people refer to an alarmone. So it will promote the cells to respond to the environment or nutritional stress. So it's like saying, well, we're running out of nutrients, right? And then what happens with cyclic AMP is that it's going to bind to a protein that is called catabolic activator protein, or CAP. And then this CAP will bind to the lac promoter, and when it does so, it will start initiating transcription. And then the cell will start using lactose instead of glucose, because glucose is gone. So here we have the growth rate of E. coli on glucose and lactose. So we have this number of cells, logarithmic 10, from 0 to high numbers, and time from 0 minutes to hours. So when you put the bacteria growing in glucose or lactose, glucose typically, it is the preferred mode of energy because it can be easily metabolized. With lactose, you have to induce your lac operon. So in this case, what you can see, it is that glucose makes the cells to grow faster in a shorter period of time versus lactose. And then when the bacteria will grow in a medium containing both glucose and lactose, then you induce this alarmone. So first you use glucose, and then you add a period of lag time in which you have a plateau because your cells won't grow anymore. They will have to adapt to form the enzymes that will make this lactose to be used. And this is happening because during this lag time, you have increased levels of cyclic AMP. So then cyclic AMP will be the alarmone that says to the cells, start building up these molecules to use lactose, which will be the lac operon, derived from the lac operon. So here we have then the positive regulation of the lac operon. So in this case, you have lac I, and then the promoter region, and then you have the operator where your RNA polymerase will bind and transcribe these LAC-Z genes, and then you will have cyclic AMP binding to this inactive cap. When it binds to inactive cap now can bind here to the cap binding site in the promoter region, and then your RNA polymerase can start inducing the rapid utilization of lactose. So how do we know this? Well, because when lactose is present, cyclic AMP levels will be high. But when lactose is absent, cyclic AMP levels low. So that means that this inactive cap cannot become active. And then, yes, you will have this cap site not bound to by this active cyclic catabolite active protein, and then RNA polymerase will try to bind, but it's not going to be induced to bind as easily as when you have cyclic AMP. That can happen a little bit before you start running out of glucose. But once you run out of glucose, cyclic AMP levels goes up, and then you start positively regulating this promoter and this region in general. So we have also something that is called epigenetic control, in which you will have additional molecules or factors that can regulate the transcription, including adding metal molecules into DNA, so that you can turn off these genes. Initially, that's what happens to the offspring. It will receive a copy of methylated DNA. That's how we know the difference between the old DNA copy or strand and the new DNA strand. The new DNA strand is always methylated, and then with these methyl groups, you have a negative charge that prevents part of these enzymes that have also a negative charge in their amino acids to go into bind into the promoter region. So in addition, at least in eukaryotes, what happens is, because we have histones which are positively charged, and if you add the metal groups, that will attract to each other, wind up much strongly the DNA, the chromatin, and then this chromatin won't be transcribed. So you need to lose these negative charges in order to have transcription. Now, this is not permanent. Otherwise, I mean, you will never have semi-conservative replication of the DNA, and you never have transcription. So this one, essentially, with time, will be lost. And then you have also something that is called post-transcriptional control, in which you will have these stop proteins that will be transcribed, and this will be essential for the downregulation of transcription. We have ribose switches, which is part of the messenger RNA, that will bind to these molecules that are called microRNAs. So these microRNAs are important to destroy messenger RNA that it doesn't need to be transcribed. So then this is how this process of microRNAs happens. So we will have here a gene segment, and then you will have transcription of these microswitches, the microRNA, and then this microRNA, when it's translated, it will bind to these regions of the messenger RNA, and then your messenger RNA will be targeted for destruction. So this is what we call sometimes gene silencing. Okay, question number 1. What is the role of cyclic AMP in regulating gene expression?
Binds to the protein, and it's in the presence of glucose, not any absence of glucose, and it pushes sort of the lacto operon to the metabolic factors.
Very good. And then next question, how does microRNA stop protein synthesis?
Complementary base pairs to the mRNA, and it creates a double-stranded region of the RNA, which uses the cell to degrade it and
You sound better than me. Come here and teach. Okay, now let's look at some changes in the DNA. So we're going to classify mutations by type, and we're going to see 2 mechanisms for repairing mutations. We're going to see what is the effect of mutagens on the mutation rate, and we're going to see direct and indirect selection of mutants, and we're going to see in a specific test that is called AMIS test, that it is used to find some mutations. So, changes in DNA results in genetic variations that can impact microbial function. Most of the times, these mutations provide an advantage rather than a disadvantage. So, the survival and the reproduction of a microbe with a new genotype can be favored by either natural or man-made environment. So basically, we can induce as well mutations in the DNA of microbes to have a benefit. And then when naturally occurs is known as natural selection, which basically selects for the best mutations. So you have the survival of the fittest in this case. So a mutation will be a permanent change in the base sequence of DNA in the code. And then this DNA will have a product encoded by this gene, and depending on what happened to this product is whether you have a mutation that might be neutral, beneficial, or harmful. So any agent that causes a mutation is called a mutagen. And then sometimes mutations happens spontaneously without having a mutagen, and this will be sometimes because these enzymes are sloppy. They're supposed to have proofreading ability, but I mean, they just plainly don't do their work. So mutations that are neutral, that it doesn't have any effect, are known as silent mutations, and this will be because it happens in the third part of the codon, the third base, which is wobbled, and then you may end up with the same amino acid or an amino acid that has similar properties to the one that you substituted. So the types of mutations that we can have, it can be a single base pair change. In this case, you can call it point mutation. And then the messenger RNA will carry this incorrect base, and then it will be read by the transfer RNA. And in these point mutations, if you change the amino acid, then you will bring the new amino acid into the polypeptide chain. But if it's a point mutation that it doesn't affect the reading for a new amino acid, they say it will be the same amino acid. And in this case will be neutral. So point mutations can be neutral, but also can be lethal, depending on what happens. So if this amino acid changes to another one, or if it changes to a stop codon like UAA, UGA, or UAG, in this protein it is essential. Like let's say that you switch this amino acid for using lactose, for instance. In this case, if you don't have this enzyme, the bacteria won't be able to survive when it is exposed to this lactose. So here is what happened with some base substitutions. So you have your replication, and then in this case you have a substitution of a G for a C for a T, sorry, a C for a T, which of course we know that you're supposed to have a C there because you have a G here in this parental DNA. And then the other daughter cell that received the correct base pairing, it is coding in this case for cysteine, 2 cysteines, but in this case, this cysteine was substituted for a tyrosine. because you change, in this case, this middle base. And then that can have an effect. Missense will be when actually you change your amino acid because you have mutation in your DNA for something that it doesn't correspond to the function of that amino acid. So that's not a neutral mutation. So you should have a change in amino acid. That's a missense mutation. So here we have another type of mutation. In this case, it's coding for a stop codon in here in this region, but before this, you have a glycine. Glycine, it is coded by GGC, and then when you change this, in this case for C, for cytosine, you change for a thymine, your your messenger RNA is going to be AGC, and you will code for a serine. So this is a missense mutation because it's not the same as the previous one in terms of what they're supposed to do. And in nonsense, you have a stop codon. So you have base substitution that leads to a stop codon. So the stop codon in here, we have it at the end of this strand, which is after glycine. But this lysine that you have here can have a change from an A to a uracil, because you have a change from a thymine to an adenine, and then you have the stop codon just right after methionine. So in this case, you have a truncated protein. And again, it can be an essential protein for the bacteria that makes it utilize nutrients, and then in that case, the bacteria can die. So, and this happens also in eukaryotic cells. I mean, people can die by a single point mutation. In this case, see, it's a point mutation. So entire organisms can die based on a point mutation. That in this case was nonsense, a stop codon. Frame shift. Frame shift is the insertion or deletion of one or more nucleotide pairs and affects the downstream region that you're reading. So you end up with a complete different set of amino acids in general, so a completely set of proteins that what you're supposed to have. So if we continue with this previous example, so we're supposed to have originally methionine, lysine, phenylalanine, glycine, and stop codon after glycine. And then in this case, we have a deletion. So you have the absence of an A, and that will change downstream here your amino acids after lysine. So instead of having phenylalanine, you have leucine, and instead of having glycine, you have alanine. And then in this case, you don't affect here the stop codon. I mean, it's a short example, but let's say that you also change that stop codon. Instead of having a stop codon, you may have something that makes your proteins to keep running, and you have a very long protein. So that's also bad. So that's frame shift. Okay, so how mutation can be beneficial? Yes, it can make you survive better, right? And I mean, thankfully, most of the mutations that we have are neutral, or they don't cause any harm to us. But in essence, any mutation that it is not beneficial because codes for a frame shift mutation or a nonsense, it can have a deleterious effect. Now let's talk about a specific mutagens. So we can have chemical and physical mutagens. So for chemical mutagens, they can directly or indirectly cause mutations. Examples, we have nitrous acid that causes adenine to bind to cytosine instead of thymine. And then we have nucleoside analogs that are man-made, like we use, for instance, drugs that are nucleoside analogs to stop viral replication. And then we have also some toxins derived from fungi, like aflatoxins, that are actually frame shift mutagens that can cause deletions and insertions. And these aflatoxins are known to cause liver cancer. So now when it comes to nucleoside analogs, we'll see in the last chapter an example of this, but basically it looks so similar to the normal base that the DNA polymerase can assimilate it, trying to put it into these strands that it's creating, but it will stop the replication of the base pairs. So we have here also an example. We have oxidation of nucleotides that makes a mutagen. So in this case, we have adenosine nucleoside, and this one is a nucleoside because it still doesn't have the phosphate group, which will make it a nucleotide, right? And then when you add this HNO2, this will bind into these molecules, and then it will steal part of these molecules in here. So we'll be altering these hydrogen bonds between the nitrogen and the cytosine nucleotide, and then you will have an oxidation of a nucleotide, that it is not useful for the DNA polymerase. So this nitrous oxide, then when it binds to this molecule, the normal parent DNA will alter. So this adenine that it is in here will bind to cytosine instead of thymine. And then this cytosine that is going to be found in here, now it will bind to guanine. And then this adenine again will bind to cytosine because it will be a permanent mutation. So either further generations will carry this. So this nitrous acid then can damage permanently future generations. So nucleoside analogs, well, we have here adenine nucleoside, and then we have this 2-aminopurine nucleoside that it has this amino group here. And then when you have this thymine nucleoside and you have this 5-bromouracil, which is the analog of this, your thymine, which normally binds to adenine, it won't be able to bind because now it will become a GC pair because this one will act like a guanine and a cytosine. Now, this 5-bromouracil, it is commonly used in labs to induce mutagenesis for studying what happens with mutations. Now, a physical mutagen, radiation. So we have ionizing radiation in which you will have two molecules that are commonly used as physical agents for producing mutagenesis. We have X-rays and gamma rays. So they will oxidize the nucleotides in DNA, causing breaks in the DNA. And then, at least in the case of X-rays, they are used as part of this treatment for people who has cancer, right? Radiotherapy. So you're trying to break down this DNA from these molecules, from these cancer cells. And then we have another type of radiation, UV light radiation, which causes thymine dimers. So two thymines binding together instead of A's and T's. That this will prevent the replication and transcription of DNA. So we have sometimes enzymes that can repair this damage by radiation. So we have these photolysis that uses light to separate these thymine dimers. And then we have nucleotide excision repair, which are enzymes that can basically cut and paste. So they cut away whatever is mutated, and then they put the right nucleotides. Not all the bacteria can do that. Some bacteria has specific enzymes, and of course, that can do this cut and paste, but the polymerases themselves, DNA polymerase, RNA polymerase, sometimes they can have integrated this nucleotide excision repair. But there are some bacteria that live in the environment, that live in extreme environments, like Deinococcus, for instance, radiodurans, that has these enzymes that makes them very fit, so they survive radiation. And there has been studies, there are researchers who does study these enzymes. These enzymes are called recombinases, Rec A, Rec B, and they try to see if one of these enzymes can be useful for us, like to swap the mutated genes that we may have. So here we have UV creating thymine dimers, and then this can be excised, and then you can repair with, for instance, one of these photolysases. There are differences in the rate of mutation, and this is something that it is well known. So we have mutation rate, which is the probability or the chances that a gene will mutate when the cell divides. And then the spontaneous mutation rate that we have is 1 in 10 to the 9th, which is kind of good. And the replicated base pairs can range as well, sometimes carrying mutations up to 1 in 10 to the 6th. sometimes we can have mutagens that can increase the chances of having mutations up to 10 times to 1,000 times. And that is bad. Now, if an organism has this mutation rate, of course, it will be much easier for that to survive than with this mutation rate. However, if this organism that has a higher mutation rate is not exposed to something that is going to mutate its genes, like it's not exposed to a mutagen, but this other one who has a lower mutation rate, it is exposed to a mutagen, then the chances are that that one that had the good genes is not going to survive as the other one. So environmental factors play a role. And I mean, major organisms like us, like humans, unfortunately, we play around with genes so much. We believe like we are indestructible, exposed so much to certain mutagens, nothing's going to happen to me. I don't wear a respirator, or I do these things, and then the consequences come. But why your grandparents were disease-free? Well, there you go. Okay, so check your understanding. How mutations can be repaired?
By like nucleotide excision repair and enzymes that come in and cut out the section that is wrong and replace it with normal DNA.
For amyloidosis that use the light for the family donors.
Yes. And then in the case of us, we have checkpoints in our animal cells, right? That prevents these mutations from being hopefully, carried to the new generations. And then how does mutagens affect the mutation rate? Increase the mutation rate. Okay, now for identifying mutants. So we have a positive or direct selection and a negative or indirect selection. So in the positive, you will detect the mutant cells because they grow or appear different from the unmutated cells. So that's positive selection. You know something. Maybe there's a change in color. Maybe you have a colony of different shape or different size. So that's positive selection. Now in negative, you detect the mutant cells that cannot grow or perform a certain function. Well, if you know that you have a mutation that affects the ability for bacteria to use lactose, you can try to grow the bacteria in a lactose-rich media, and then you will be selecting for the ones that do not grow there, because that's an indirect way. And then we have an example here of auxotrophs. These will be mutants that has a nutritional requirement that is absent in the parent. So you, in this case, had originally a bacteria that it was lactose positive, so you can use lactose, and then you mutated your bacteria, and then you created a lactose minus. So this one cannot utilize, so that's an auxotroph. And then you can use replica plating to find out where is your mutation. So you have, in this case, a media, a Petri plate that has histidine, this amino acid. And then you have this device that has this circular surface that it is in similar size, circular, to this Petri plate. And then you wrap around within this device kind of a fabric that is made out of velvet that is sterilized. And then, like if it was a stamp, you put it on top of your PG plate that has colonies growing in this master plate that can use histidine. And then you will have two different types of media. One that it has histidine, and that one that it doesn't have histidine. So then you stamp in your new plates without bacteria your replicate from this master plate. So you stamp it both, and then you grow it for 24 hours. And then you will find that some of these bacteria, which are the mutants, the auxotrophic bacteria, won't be growing in this media that is lacking histidine. So that's your selection, your indirect selection. Now, for identifying chemical carcinogens, we have this test that basically help us to test mutagens using bacteria as a carcinogen indicator. This is called the Ames test. So in your Ames test, you expose your mutant bacteria to substances that can cause, in this case, cancer. So these mutant bacteria, when you expose it to these mutagenic substances, you measure the rate of the reversal of the mutation. So initially, for instance, you would have a media that that it is rich in histidine. And then you will have a bacteria like Salmonella, which is an auxotroph. It cannot utilize histidine. And then this bacteria, you expose it to a mutagen. And then you will see if this bacteria would, with time, can utilize histidine to become histidine positive after exposure. So if this bacteria will reverse in a specific time versus other ones, you will be measuring the reversal of the mutation. In this other example, you have a rat liver extract that it was added to provide activation of enzymes, since this animal enzyme sometimes can facilitate mutation. And then if these enzymes will be utilized to certain, will be able to help for certain organisms to reverse to the utilization of certain nutrients, because these enzymes code for the utilization, for instance, of glucose. In this case, you will have this positive test. So the reversion rate indicates the degree to which a substance can be mutagenic eventually. So here we have this example. Two cultures are prepared of Salmonella bacteria, in that they have lost the ability to synthesize this histidine, so they will become histidine dependent. So you have your experimental sample. When you put your bacteria together with the suspected mutagen and your rat liver extract, and then you have your media lacking histidine here, you incubate it, and then you have your experimental plate. Now, in this control, you just use the rat liver extract, but you don't have, and your bacteria, of course, but you don't have this mutagen. And then you put this part of this media into your plate, but after you incubate it, you have only few colonies. The colonies that were able to overcome this mutation. So in this case, you would have a positive Ames test showing that you can have a great mutation. So how would you isolate an antibiotic bacteria that is resistant to a particular antibiotic?
Do replica plating and the velvet stamp and plates with the test the antibiotic on those replicas.
And what about how do you isolate an antibiotic sensitive bacteria?
So methods the antibiotic, the replica plating and put antibiotic in plates and see which ones the colonies are missing from. And the colonies to do one with each one and antibiotic.
So you can do replica plating in both, and positive selection will be selecting for these ones that look different. And then the negative will be for the ones that actually do not grow. So that's the indirect. So how will that play a role into this? Which one will be the indirect and which one will be the direct?
I think antibiotic resistant would be indirect. Sensitive, actually antibiotic sensitive is indirect, and then the first one, antibiotic resistant would be direct.
Mm-hmm. And then what is the principle behind Amy's test?
The potential mutagen bacteria and it's causing the reversal of that for like the histone. The bacteria is can't produce its own histidine, so it reverses it and makes it produce its own histidine, and it can grow in cultures without histidine.
Yes, very good. Okay, now we're going to look into plasmids and transposons, and we're going to see the difference between horizontal vertical transfer, and we're going to look at the methods for genetic recombination in bacteria. So genetic recombination itself is basically making a hybrid. You have exchange of genes between two DNA molecules, and then you have a new combination, and generally speaking, that produces diversity, genetic diversity. And that can happen in prokaryotes and eukaryotes. Now, in eukaryotic cells, we have this specific mode of recombination that we know as crossing over that happens during meiosis, in which you have to have physical contact between chromosomes of two different parents and exchange of these chromosomes. And with time, we have, of course, this set of gametes that basically makes us unique. I mean, how we end up sometimes with the hands, the eye color, or certain characteristic of one of our grandparents. Well, it's because of this type of genetic recombination, the crossing over. So you have a donor DNA, and then you have a recipient DNA in this genetic recombination crossing over. And again, you have a physical connection between donor and recipient, And then in this case, we have recombinase A protein that swaps pieces. So in this case, this donor and this recipient will exchange these genes. So they are not the same, at least in that strand. Now we have vertical transfer and horizontal transfer. Vertical transfer will be transfer of genes from an organism to its offspring. And then horizontal will be an F plus and an F minus exchanging genetic material, which is basically, in this case, will be a plasmid transfer. And the donor cell will be the F plus and the recipient will be an F minus. And after you have this transfer, the F minus will become F plus. And this one is very common because it will help bacteria that weren't able to maybe utilize a nutrient or to tolerate the presence of an antibiotic or a disinfectant to now be able to do so. So it's very common in the prokaryotic world to have horizontal transfer. And then we have pieces of DNA that exist outside of these chromosomes. We already know about the plasmid DNA, but we have mobile genetic elements. So they can jump between chromosomes or from one cell to another one. And it could occur both in prokaryotic and eukaryotic organisms. In terms of plasmids, they are self-replicating. They're small circular pieces of DNA and makes between 1 to 5% of the entire genome of the bacteria. It is found primarily in bacteria, and it will encode for proteins that increases the resistance of the bacteria in a media, or sometimes it makes the bacteria to produce toxins. So these genes that cause for resistance are known as R factors. And then you can have several of them combined into a single region. This is called resistance transfer factor. And they help for replicating or conjugating. And then you have the R determinant resistant genes. When you work with these plasmids, there are certain software programs that tells you the exact nucleotide bases that exist within this plasmid DNA, and it tells you as well where you have a specific sequences, like the origin of replication in here. That's where you start actually making another copy, not everywhere else. So that's why it's called the origin of replication. And then we have here the R determinants. So basically, this is what this DNA makes the bacteria to be able to do. So for instance, we have mercury resistance gene here, sulfonide resistance gene here, streptomycin resistance here, chloramphenicol resistance, tetracycline resistance. And then on this region, we have the origin of transfer. So basically, that's where the bacteria starts transferring the DNA through the pilus. And then we have the pilus and conjugation proteins here. And this will be the RTF, which is the resistant transfer factor. So this is what makes the bacteria to be able to transfer the plasmid DNA. So this is like functional genes, while these ones will be structural genes. This is what makes the bacteria to be resistant to several to several antibiotics or substances. So we have different types of plasmids. We have a plasmid that is called conjugated plasmid that carries genes for sex pili and transfer of the plasmid. And then we have this dissimilation plasmids that encodes for enzymes that make some bacteria to use unusual compounds. And then we have the R factors that encodes antibiotic resistance. And then in the case of the previous example, we saw multiple antibiotics, including tetracycline, chloramphenicol resistance, which will be together with this one, the mercury resistance. Mercury resistance in this case will be dissimilation, and then the other ones will be R factors. Transposable elements or transposons are segments of DNA that can go from one region to the next. These are also known as jumping genes. And they have IS sequences, insertion sequences. So these basically have an enzyme that makes cuts within this area where they want to insert, and to be able to reseal that DNA. So it's like the copy paste. And may inactivate genes if they insert within genes. Well, in the case of certain transposons, they can also activate genes, but they can inactivate genes. And depending where they insert, they can have a beneficial or a deleterious effect. Like, let's say that this transposable element binds into a region where, in the case of eukaryotic cells, you have tumor suppressor genes and inactivate the tumor suppressor genes. In this case, this cell will be undergoing several mitosis rounds, and then it will create a tumor. So that's one bad thing that it can do. And you have class 1 transposons, which are basically transcribed forming RNA, and then they can make a reverse transcriptase enzyme, and then they can insert into the DNA. And then you can have class 2, that they don't need any enzymes to insert into the DNA, like in the case of class 1. And then you have a complex transposons that can carry other genes, like enterotoxin production, antibiotic resistance genes. It's just like they carry along themselves. When they insert into certain organisms or certain cells, they can carry these resistance genes and put it into another one. So we have here a class 2 transposon and insertion. So we have inverted repeats within this transposable element, and then you have your transposase gene here. And all of this is an insertion sequence. So these inverted repeats that are found at the ends of this are, just as you can see, they are inverted. So this will be the inverse of this. Like this A will be here, and then you have these T's in here, and these A's in here. So when you have transposase, this transposase will excise these two pieces, and then this new element without these two pieces will be able to bind into a region of the donor, the recipient, that is actually having complementarity, like in this case. So you can copy and paste within that region. Okay, what type of genes do plasmid carry?
Antibiotic resistant genes and genes that bacteria to use unusual nutrients like the mercury dissimilation genes.
Dissimilation genes, yeah. Okay, now let's talk about transformation. So, transformation itself is basically the transfer of genes from one bacteria to another one. This is what sometimes happens, and that transformation will be specifically within the DNA itself, like the nucleoid DNA. So here is an experiment that was done in the 1960s that helped to understand that actually DNA was the material that it was transferring genes. Before that, they were debating whether it was proteins that were the genetic material and not DNA. So in this case, we had two bacteria, one that had the ability to make, and this bacteria was Streptococcus pneumoniae, one bacteria that was having the ability to create a capsule within this bacteria, and another one that it didn't have the ability to create a capsule. In this case, this bacteria, when you grow it in the lab separately, like the one with capsule and without capsule, they look differently in the Petri plates. And then they combine the two bacteria, the one that codes for a capsule and the one that didn't code with the capsule, together. And then eventually, these bacteria transfer DNA into the one that didn't have the ability to produce the capsule. So that's transformation. So they injected this new bacteria into the mice, and then the mice died. And then they extract part of the blood of the mice, and they grew the colonies. And they saw that the mice died because he was having this encapsulated bacteria in his bloodstream. And then the bacteria, it looked like the original encapsulated bacteria. Now, whenever they injected only this bacteria in a mouse that it didn't have a capsule, the mouse remained healthy, it didn't die. And they grew the bacteria from the blood of these mice, and then they saw the colonies that looked like the non-encapsulated bacteria. So then they also, what they did is they heat kill this bacteria that had the capsule, and then they injected into the mouse. And then the mouse remained healthy. And then no colonies were formed because they heat killed the bacteria. So they cultivated the blood into a Petri plate, and nothing was found. Now, in the next generation, they had the living non-encapsulated bacteria and the heat-killed bacteria injected. In this case, will be the heat-killed encapsulated bacteria injected into the mouse, and then the mouse died. And then this bacteria that was the living non-encapsulated actually got the DNA from the heat-killed bacteria, and then it was transformed. And then when this mouse died, they cultured the blood in this petri plate, and the colonies looked like this one, meaning that the DNA from this heat-killed bacteria, that it was the encapsulated bacteria, wasn't destroyed by the heat killing, and then these bacteria that non-encapsulated get this DNA, got transformed, and then this DNA code for the capsule, and then this bacteria, which is more aggressive than the non-encapsulated, make this mouse to die. So that's how they prove that actually DNA was the actual genetic material and not protein, as they believed before that. So, sorry for